Level 3 and 4 can be really tricky, mostly level 3. In level 3, you're introduced to a concept called epistasis. And epistasis is just this idea that some traits turn on other genes. So certain genes turn on other genes. For example, the color gene will influence whether or not some other genes show up. In this case, if you have the color gene, then you have the possibility of having gray or orange. But without that color gene, you've got no possibility of having the gray trait or the orange one. So you got to pay close attention to those things. The other thing that's really emphasized in this level is the idea of what's called the central dogma of biology. That is, DNA determines RNA, determines protein. So the DNA will determine the protein. Protein is going to determine traits. In this case, the trait is color, the protein, melanin, that's skin color, and you're going to have proteins that are in charge of making the melanin. So the whole chain has to be working in order for you to get the trait that you're looking for. This chart is going to be super helpful. So pay close attention to this chart. The things on this chart are going to be significant to solving that riddle. You'll notice you have the melopod, that's the cell that contains the skin pigment. You have the melomaker, looks like gears. Those gears are going to be making the skin pigment. You have the melohelper. The helper is keeping the maker together. All these pieces have to be working in order for you to get the traits you want. If that cell grows really big, you're going to have a gray dragon. If it starts to get smaller, you're going to have an orange dragon. If it's really small, you're going to have an albino dragon. So in order for me to get that cell to be super large, I need the melanin maker plus the helper to keep that complex together. When that happens, I'm going to be putting those molecules together, making lots of melanin. If the melohelper is not functional, the makers aren't going to be together, then the cell is going to start to get smaller because it's not going to be making and storing very much melanin. So I need to be aware of that based on what the challenge is for me. Ultimately, I'm going to get a chance to manipulate the genes. The genes determine the proteins determine the colors. So when I get to manipulate the genes, I want the genes to have or to code for the traits I'm looking for that will give me the colors that I want. If I have the functional maker and helper, I'm going to have a large cell. If I have the helper that's not functional, that cell size is going to get smaller. It's going to change the dragon's color. Now we have another option. The albino option might be because the melanin maker is broke. Remember, proteins are like tools. If we somehow mess that up, or if that protein isn't working right, then it's not going to be making melanin like it's supposed to. So you'll notice now I could have the makers wrong. I could have the helper as wrong. That's going to give me an albino dragon. So again, these are things you're going to want to be aware of. How does this process work in order to get the dragon color I'm looking for? Ultimately, again, I'm going to be able to manipulate the genes. When I get to manipulate the genes, I'm going to be able to influence that process. When I influence that process, I'm going to be able to get the colored dragon that I'm looking for. Here's another example of how protein function matters. So here are two types of proteins. These proteins allow the melanin to be released to turn the dragon different colors. I have a functional one and I have a broken one. So study this chart. That's going to be helpful. Whenever I'm getting the melanin pods like moving through the cell membrane, I can destroy those, or I can bump them back or manipulate how they move. And I found that if I keep them out of the tunnel at all, that's the best way to get the color change as quickly as possible. Otherwise, they're going to be released or not, depending on what kind of protein is in that channel or in that gate. Pay close attention to those things. Don't be afraid to experiment a little bit. Record what's happening. Get that process down. Keep trying because science is tricky, and don't give up. Apply those principles